Start recording. Hi, and welcome to the webinar titled Cultivating Soil Health in Orchards, What Really Works. I'm Sue Chen. I'm on the advisory board of Oregon Organic Hazelnuts. I'm also a farmer of a certified organic hazelnut orchard and beekeeper. So I love soil, especially healthy soil. And so, for you voracious note takers out there, just so you know, this is being recorded, and we will also be following up with everyone with that recording and all of the resources. So, first off, I just want to thank you for making this investment of your time, especially you farmers out there, because this is a busy time of year, to be on this exciting webinar and really a very important topic. We have a fantastic turnout today, which is so encouraging. And I also want to thank everyone who registered early and submitted excellent questions. We were able to get them early in the process. And so for you guys who signed up later, we'll have ways that you can engage with questions, and Shannon will be sharing that. And we'll be addressing many of those questions that you did submit in this webinar. So I'm thrilled to introduce our contributors today. First, we have Penny Feltner. She's the program manager for OrCAN, that's Oregon Climate and Agricultural Network, Soil Health Network, which is an inclusive, collaborative statewide soil health network to promote soil health for climate resilience with a focus on meeting the needs of historically marginalized and under-resourced producers. She has over 15 years of experience in the agriculture and environment sector with a focus on agroecosystems, climate change adaptation, and program development. She earned her BA from Thomas More University in Kentucky and her master's in environmental science from Miami University in Ohio. She's worked with farmers on a variety of environmental initiatives in the American Midwest, North Africa, and the Caribbean before settling in the beautiful state of Oregon. Welcome, Penny. And Dr. Shannon Capellazzi is a soil health extension specialist in Oregon State University's new organic ag program. Growing up in Oregon and having 3 degrees from OSU, she has her roots in this community and a strong connection to her cattle ranching background. She brings a depth of experience in soil health management and quantification, having held roles at Go Seed, Soil Health Institute, OSU Central Analytical Laboratory, and Wilbur Ellis. She will share the knowledge gained through her experience from projects using cover crops in hazelnut orchards. Welcome, Shannon. And so I'm going to prime and prep you with the webinar objectives today. They are to increase understanding and appreciation of soil functions in orchard systems, introduce orchard-specific applications of soil health principles, also address common perceived barriers and clarify common misunderstandings regarding soil health, and then also orient you, our attendees, to support resources for implementing soil health practices. All right, let's get started. I'm going to turn it over to you, Shannon.
All right. Let me share my screen here. All right, let me move that one. How we look? All right. Thank you for all of you for joining. I saw quite a few familiar faces out there, so welcome friends and folks we haven't met yet. I hope this is the beginning of our conversation. I also wanted to just thank Sue and Penny for all of their work in coordinating this and for really kind of honing in on the what works part. If you've ever heard me talk about soil health before, I often talk about functions, and that's getting the soil to work. And so, this title just invokes in me like thinking about the soil doing work. I already had an introduction, so we're gonna dive right in. And just an overview for what we're going to do today. I plan to weave the questions that we got ahead of time kind of throughout each of these topics because they really fit into a lot of the things that I was planning to discuss. So we'll start with soil health and what those soil functions are, management practices that influence soil health, how soil's inherent capacity influences, particularly interpretation, a little bit about soil biology and the organisms and communities in there, and then hopefully have just a little bit of time at the end for some discussion. So I know everybody's on Zoom. You don't really have to close your eyes, but I think that it helps as we do this. So go ahead and close your eyes. Take a big deep breath. Let yourself relax for a second. And I want you to think about the best smelling soil you've ever experienced. As you have your eyes closed and you're thinking about what that soil smells like, I want you to think about what it looks like. What's that pore structure? What's the color? What's the space in between those mineral particles? What are the plants that are growing above it? What kind of animals or organisms might you see? How does it feel? How does it smell? And have you ever tasted it? This is something that I do often with people when we're in the room together, and I love to have people share their thoughts. But today we're going to let you just kind of hold on to that. And the reason that I start with this is because I want you to know that you know what healthy soil is. When you have felt and smelled and you've seen that soil that looks really healthy, you know it. You feel it in your bones. But it can be a little bit hard to get yourself into the right perspective. So I have this picture here on the bottom of the screen of a cave that I was in. And I just remember thinking to myself, this is what it feels like to be a microbe in a soil pore. As I'm walking through this cave system, I was like, oh, there's these different spaces and there's all these different textures and there's minerals kind of moving this way and that. And there's areas where the light hits and there's green and there's more water and less water. And, sometimes it's hard to think about that perspective because their whole world is in a 1 millimeter space. So I think about being in a cave and I think about, trying to evoke that. So I just want to let you know, you all know what healthy soil is already. We're going to talk today a little bit more about how we get that healthy soil. Um, and I also do want to give you that definition of soil health. Soil scientists have given us a good definition, I think, and I come back to it over and over again because it grounds me in how I do evaluations and how we aim to improve soil health. And so soil health is the continued capacity of the soil to function as a vital living ecosystem that sustains life. There's a lot of different iterations of that. That's kind of my simplistic version of it. But I key in on the fact that it's all about that soil functioning and that it doesn't do that unless it is that vital living ecosystem and that everybody on this planet depends on the soil's ability to do that work. So, digging in a little further, we're going to talk a little bit about the functions of soil. Again, live in person, I might have you shouting out what these different functions of soil is, and you can all tell me all the things you remember from your Soils 205 class, but we'll go through these a little bit differently. There are major functions. I think one of the first things people think about are related to soil functions with water. We need the water to be able to flow into the soil. That's that water infiltration. Right? We need the water to go in. We don't want a sealed surface with the water running off. And so we want water to flow in. We also want that water to be held. We also want that water to flow through, and we also want that water to be filtered. So that's asking a lot of different things out of one kind of medium. And that soil has a huge influence on the entire global water dynamics. We need that water flowing in so that it can be held by the soil, so that it can be filtered by the organisms and the structures that the organisms made. We need that water to move through and be replenished. Similarly, we need some of the same things for the nutrient cycling, right? Nutrients are coming into the soil from a variety of different means, and we need those nutrients to be transformed from maybe a dead plant organism or dead or poop or whatever it is. Those nutrients need to be cycled by the soil. And when we say something is done by the soil, most of the time it's done by the organisms that are living in the soil. A lot of the nutrient cycling processes are biologically mediated. That means there's some little organism in there that is excluding an enzyme or an acid. And as they excrete these different chemicals, it changes the nutrient availability. We take larger organic molecules, break them apart so that plants can get the nutrients and continue that nutrient cycling process. Along with that, soil is just considered a habitat. It is the most diverse habitat anywhere on the earth. There's a really cool, large paper done that just came out with this result. Soil is the most diverse habitat, and over 60% of all of the organisms anywhere in the entire world exist in the soil. It is a habitat in and of itself, and often when I think about soil health, I'm thinking about it as that habitat. What is required for any organism to survive and to have a good habitat? They need food, they need shelter, they need water, and that's what the soil can provide when we take care of it as a habitat for those organisms. Um, Another major function of soil is as a gas regulator. Microbes, a lot of microbes take carbon into their body, and their byproduct is CO2. Just as we exhale carbon dioxide, that CO2, a lot of organisms also exhale carbon dioxide. Plants, we know, take that carbon through photosynthesis, fix it into their body, and they send a lot of that carbon down through their roots and into the soil. There's a wide range depending on a whole bunch of different factors, but somewhere between 20 and 60% of the carbon that's fixed through photosynthesis in the plant is sent down through the roots, and a lot of that is going out of the roots to feed the organisms. There's no way the plant would do that unless that was economically efficient in plant terms. And so they're taking this carbon, they're using their energy through photosynthesis, sending it down through the roots and feeding those organisms. Again, providing for that habitat and allowing for nutrient cycling and water storage and all of these other components. Those things together um, make this medium for plant growth. Sometimes we think about this as that structural stability. It just needs to have a space where the roots can move through, and those roots can be solid enough that when the wind comes through, it's not going to move them over. But when we call it a medium for plant growth, we might consider all of these functions of soil as part of that story for that medium for plant growth. Uh, Somehow nutrient storage got over on the other side of this figure instead of being right next to nutrient cycling. But we talk about nutrient cycling. We also need that soil to hold on to nutrients. There's a ton of nutrients that are in the soil that aren't always necessarily plant available. They're stored there. They go through a process and eventually become, they can eventually become plant available, but there's a lot of nutrients just sitting there ready for microbial action. And then the last thing that we think about when it comes to functions of soil is uh, soil as an engineering medium and something that we build on. It's something that we build with. It's something that can help us design where our water is moving. It's something that we need to consider. The characteristics of the soil are things we need to consider when we're planning where our barn is going to be or where we're putting a livestock path or how we do how we set up an orchard. You can call it 5 functions of soil. You can call it 8 functions of soil. You can call it 21 functions of soil. But the idea is to think about all of the things that we ask the soil to do for us. All of these things are ecosystem services, things that the world does for us that help us and allow us to do the rest of our lives. So how does it do this? Soil doesn't do this on by itself. Soil needs plants in order to provide these services. They work together. You don't have soil without really, without any plants there because otherwise you just have minerals. Once you have plants, you start to develop that soil. We're not going to get too much into soil development here, but some of the things that plants do is that they're protecting that soil surface. When a raindrop falls from the sky and it hits a bare soil, it dislodges all of these little soil particles and they blast all over the place. When you have a big, healthy plant biomass over top of it, it hits the plant and it trickles down to that soil surface, which is going to have a huge influence on decreasing erosion. The plants are also going to help to moderate temperature and moisture. And this, I think, is one of those fundamental biological concepts that we need to think about, and that is that temperature and moisture influence the rate and activity of almost all of any organism. We have the ability to moderate our own temperature, but most organisms don't. And moisture and the amount of water that we have available is going to influence every single organism and their ability to do work. And so having the plants going to moderate that temperature and moisture in the soil. They also create these flow pathways. We talked about the importance of soil being able to infiltrate and flow water through there. Roots create pathways, they create pores, they create ways for that water to move into the soil. And then one I think that we talk about a lot are these plant roots exuding carbon into the soil. And and that does a couple of things. Number one is that it is feeding microorganisms. A lot of the microorganisms in the soil take carbon into their body for their energy. They take that carbon in and they exhale that carbon dioxide. Most of the carbon that they're taking in are exudates from those plant roots. And so we need those plant roots to actively be taking carbon out of the atmosphere, pumping it down into the soil to feed those microorganisms. The other thing is I would say that soil functions absolutely rely on microbes. And this is everything from bacteria and fungi to archaea and insects and, well, they're not microbes, sorry. There's a lot of different kinds of biology in the soil. Microbes do a lot of the day-to-day work, but there are other organisms who all play a role as well. So maybe I should change that to rely on soil organisms. But we will talk about microbes a little bit here. One of the things that we know is that there are whole classes of organisms that take nitrogen from the atmosphere. Most of our atmosphere, maybe 78% or well, about 78% is made out of nitrogen. It is dinitrogen, two nitrogen molecules bonded together really tightly. They've got three bonds in there and they don't like to separate. Rhizobia bacteria, which associate with legumes, have the ability to break apart that bond and make that nitrogen available to plants. That is a huge service. Nitrogen is necessary for every kind of protein and amino acid, and it is one of those things we think of as the cellular building blocks of life. This is one of those things that we need absolutely, and is often limiting our plant growth. And so having those rhizobia bacteria associated with legume plants allows us to make more nitrogen available to the plants. Another thing that they do, we think about, oh, and let me just, can you see my pointer on here on my screen? Can you see that? I see some heads nodding. These little nodes right here, these are nodules. Those nodules are where the rhizobia that are associated with legumes live. And so if you have something like a clover or any kind of bean or vetch that is growing, pull it up, look at the roots and see if you can find these little nodules. You break it open and you see it as like pink or red. That means it's actively making nitrogen available for your plants. Another big thing that they do, organisms do, and this is largely mycorrhizal fungi, is that they increase the root surface area. And this is really important because most of the nutrients and water that the plants are taking up are because they come into pretty close contact with some kind of soil particle. And so if you can increase the surface area, you're increasing their ability to touch different soil particles and increasing their ability to scavenge nutrients and water. And so we can see this huge increase in the plant's ability to get nutrients and water when they have this increased root surface area. A lot of those arbuscular mycorrhizal fungi are also going to be producing acids and enzymes that may also aid in that ability to pick up nutrients. Another thing that fungi, bacteria, archaea, algae, they all do is they all play a role in building aggregates. Aggregates are conglomerations of soil particles. So when we talk about particles, we have sand, silt, and clay-sized particles, with sand as the biggest and clay as the smallest. We want them to group together because when they group together, they make kind of balls that, make larger pore spaces between these balls. So if you've got one clump here and one clump here, you're going to have some larger pore spaces. We need those large pore spaces so that the water can infiltrate and so that the water can flow. But the other thing they do is when they aggregate these soil particles into these aggregates, when they are conglomerated and glued together, it makes them really, really tiny, small pore spaces as well. And those tiny pore spaces are where we hold water and where that water can be held against gravity flow. And so building aggregates and the corresponding pore structures allows the soil to do all of those water dynamics that we really need it to. It allows that water to infiltrate and for it to be held. These glues that all these organisms exude to hold these aggregates together also play a huge role in the filtration system for water and helping that water quality component. One of the final
Shannon,
things I want to talk about with microbes really quick.
you
Am I
got
at 10
20.
minutes?
No,
20?
20, 20.
Okay. Uh, I have a note on here about chemical communication signals. There are all sorts of volatile organic compounds in the soil, in the air that are used as communication signals between all of the natural world. And it's not necessarily a conscious communication, but it's communicating stress. It's communicating drought or pest or disease or something going on. Sometimes it's communicating, hey, there's too many of this organism around here. We need to adjust our populations. Um, and this all happens through the microbial community in the soil and has a huge role in making our soil and our plants healthy. The bottom picture here, if you're looking at it, these are different soil particles. We've got some very small particles, and this kind of snot-looking stuff, this is what we call extracellular polymeric substances. I just call it microbial exudates generally. It's stuff that they exude out of their body, and it can be any kind of material they put out there, and it has a huge role in anchoring and building those aggregates, and it's also where a lot of the nutrient cycling is happening with those organisms. So we kind of got through our functions of soil here. And I wanted to jump next into the soil health principles. First, I want to say that every different production system can take these kind of principles and apply them a little bit differently. There's going to be differences in every production system and a different way of using them. But these are these overall principles that we say, when we do this, we are moving towards improving that soil microbial habitat. We're moving towards increasing that soil's ability to function using that living and natural system. And so I put first and foremost increasing live roots. Having that plant cover on the ground and exuding that carbon and feeding the organisms in there, that is the ultimate. Whenever you can increase live roots, and that's increase the amount of area that is covered by live roots, increase the amount of time of each year that live roots are active and present. That's going to help to feed that microbial community. Increasing cover. This is another one that we talk about, and this can be a live cover crop. It can be a dead mulch on the system. It can be a plastic mat. It's not always going to be the same thing, and depending on your system, it's okay to have differences, but this increased cover is going to have a huge influence on what we were talking about earlier with that water drop coming down and dislodging these soil particles and causing that soil sealing. It can also create a really nice different layer of a habitat. I have a whole talk on different bioreactors in the soil, and there is a whole habitat in that mulch layer above the soil. And so not only does it help reduce erosion, but it can also help with fostering different kinds of communities. Next one on here is to increase diversity. And so you see a picture here with some crimson clover and phacelia sitting next to some vetch and some oats. And we talk about different plant functional groups a lot of times. One of the things you'll often hear is that greater above ground diversity leads to greater below ground diversity. And there is some truth to that. And it's not exactly a direct relationship. But when we can increase that above ground diversity and we can support the microbial habitat, we do tend to see more microbial diversity underground as well. We'll talk a little bit more at the end about the importance of that. Minimizing disturbance. Here we have a big, beautiful cover crop that is being worked into the soil. There is a time and a place sometimes to get that biomass into the soil. There are also other ways of terminating a cover crop where you don't necessarily need to till it in. It's a balancing act. We don't say never disturb the soil here. There is a time and a place for some of this work, and there are other ways of disturbing the soil besides just mechanical cultivation, right? When we talk about spraying something on there or we talk about having animals on there, all of those are disturbing the system in some way. And so if it's something that changes the environment of those organisms in the soil, that is a disturbance. And so it's something to consider. The next one on here is integrating livestock. This is not required for anyone. None of these are required for anyone. Um, and it can be a real challenge. We can talk a little bit at the end about really important considerations. Integrating livestock should be done with care, and it can lead to some of the great soil health outcomes, and it can be really challenging. And so it's something to be taken on very intentionally. Um, I was able to do about a 3-year project working on cover crops in hazelnuts, partially because you see so many new acres of hazelnuts going into the Willamette Valley, and there's a lot of soil that is kept bare because of the way that they typically are going to be harvesting hazelnuts off of the ground. We had a lot of really nice success talking with producers who have used cover crops, ways that they have made it work. And here's kind of some of the key takeaways for making cover crops work in hazelnuts, where they need to be able to sweep and vacuum nuts right off of the ground. Number one, most of the producers are maintaining a clean tree row still. That doesn't mean that, that means they're increasing the live roots. They're increasing the amount of days with live plants on there, and they're cutting down on the herbicides. Um, I think one of the most important points about this is that the selection of which cover crop you use is absolutely key. You want something that's going to be able to have a nice smooth surface, and is going to be easy to keep short in the fall. You don't want it to be bunchy. You don't want something that's going to create big holes. And so we really think about that harvest feasibility when it comes to selection of the cover for hazelnuts. I highly recommend doing a thorough job of mowing or flailing all of your biomass in the middle of the season, June or July, so that there's time for most of that material to decompose and to make sure that you get a nice shortcut on it right before nutfall, because they are harvesting with sweepers and vacuums. So one of the questions directly from our audience, that's questions from the audience, was what harvest machinery do you need when you have cover growing between rows? What works with my hazelnut harvest needs? So I would say, number one, most people are using some kind of mower or a flail. Anything that you can get your hands on that's going to cut it relatively close to the ground is going to be okay. There are some folks who have attachments that blow the biomass into the tree row to make a mulch, and they really like those. Others where that biomass sits there on the ground, and you know what? It decomposes by harvest time most of the time. When you have legumes in your blend, you're going to have faster decomposition rates. And when you have a ton of biomass, you might have to really pay attention to make sure that you get that cut early enough that that's not going to be interfering with harvest. I would say on this, the mowing timing is everything. Depending on the cover crops that you have chosen, you might want to cut before or after seed maturation. That's going to depend on your goals. Do you want it to self-reseed itself and maybe go again? Are you in a perennial? So you have to think about whether you want to cut it before or after seed maturation. But I highly recommend cutting in June or July so that there's time to decompose, and then an additional mowing right before harvest. Another question from the audience, and this is something we could have an entire webinar on, and I expect that will be coming sometime soon, but that is cover crop mix options. Whenever I talk with somebody about this, the first thing I ask them is, what are your goals? What are you hoping that cover crop is going to do? Because until you know what you want from that cover crop, it's really hard to choose which species you want and how you're going to manage that. Once you have an idea about what your goals are for your cover cropping, I highly recommend going to westerncovercrops.org. There is a decision tool, which is the cover crop selection species selector, cover crop species selector. And you can find a lot of information about individual species and how they might perform in your area. For a lot of orchards, I recommend some kind of sod-forming, shade-tolerant, cool season grass. Um, the hazelnut folks have had great success with creeping red fescue, and also some with chewings or hard fescues. Tall fescue can be really nice sod forming as well, but I don't really recommend it, especially not forage tall fescue. Not recommended for hazelnut producers for what's going to happen in the fall, right before harvest. Another option is Kentucky bluegrass. And there's people who are talking about both bentgrass and poa annua as well. They're not going to be creating a lot of biomass, but they are going to keep that soil in place. They are still going to have some nice impact. Often when we talk about these cover crop mix options, we have the best results when we have some kind of combination between grass, legume, and some kind of forb. And so we have these sod forming cool season grasses, as well as either an annual or perennial legumes. Most people in orchard systems end up using a white clover in their perennial systems, and that tends to work pretty well. There are folks who also use alfalfa or red clover. Those are options. Or if you can, go out there and either broadcast or drill annuals in there. Crimson clover and Persian clover work really nicely. Subterranean clover is an interesting option because it's considered self-reseeding. Uh, it will it will self-reseed. You can cut all that biomass down in the middle of the summer after seed has been set, and then when the rains come back in the fall, it'll pop up again. Um, Common vetch and hairy vetch, I've also talked to quite a few people in orchards who like that. Again, I'd be a little hesitant on hazelnuts for the vetches, but I do know people who say they really like them in those systems. Um, Plenty of people have goals where their ideas that they want more pollinators, right? There are plenty of orchard crops where we need pollinators out there. And Phacelia is one of my favorite pollinators. It is gorgeous. The bees absolutely love it. And it has a really, really gorgeous root system. It's super drought tolerant. And then I wouldn't probably plant daisies or dandelions in my own orchard, but there's plenty of people who have told me, you know what, they're not doing any harm. They're fine out there. They're adding a little bit of diversity. Who knows exactly what they're exuding into the soil, but they seem healthy and they're not causing problems. Um, Again, for hazelnuts or other crops that you might want to be sweeping, they might cause problems. So be careful with that. But there's plenty of people who want to incorporate more flowers into their cover cropping options. This is another one that we probably could do a whole webinar on. The question, what is the cost benefit of planting cover crops?
Shannon,
And
before you go into
yeah,
that, just so you know, you have 15 minutes but that includes the questions that you're going over. So you do have
okay,
15. Okay.
okay. Next one is what is the cost benefit of planting cover crops? I want to give the caveat that this is specific for each orchard. It's really hard to give any kind of blanket statement because there's a differential response by soil type, by where you are in the landscape, by your management history, by the species of your cash crop, by the species of your cover crop. There are a lot of different things to consider. So I'm going to just leave this list here with you with some of the direct costs, some of the indirect costs, and then some of the direct economic benefits and some of the direct ecological benefits, and let you look through those or screenshot them. Um, it is something to discuss with a trusted agronomic advisor for how that's going to work for your own system and how you can optimize the benefit portion of this for you and your goals. All right, just a little palate cleanser there because there are a lot of words on those slides. These are two pictures taken on the exact same farm from research trials where we had a diverse cover crop blend right next to a no cover control. These are taken on the exact same day. One of the things people say over and over is the cover crops steal moisture. And there are some situations where there's absolutely some challenges with that. And there's also a lot of other ways in which those cover crops can help the moisture dynamics of the system. Um, so I'm gonna leave that one there. This is data from a hazelnut orchard. We were able to install temperature and moisture sensors at two different depths for an entire season at multiple locations through one orchard that had multiple treatments. This data right here that I'm presenting is from that 6-inch soil depth and just the 5 p.m. each day soil temperature. The cover crop in this situation was just Poa annua, which is, for those of you who don't know it, it's a very small grass. It is dormant in the summer. There's relatively minuscule roots. There's not a whole lot going on. In the summer, it just looks like dead brown fuzz on the soil surface. But even with that, we see a huge difference in the 6-inch soil temperature. So what we see in this figure right here, each of these bins is by air temperature. So when the air temperature was 62 to 71 degrees throughout this whole time period, we see that there's a small difference in the 6-inch soil temperature in the bare orchard floor and in the cover cropped or the Poa annua covered floor. Once we get up into these hot days where that air temperature is between 90 and 105 degrees, there's a huge difference in what that soil temperature is. On average, it was about 20 to 25 degrees difference at that 6-inch soil temperature. That's going to have an influence on evaporation. It's going to have an influence on your microbes' capacity to function. It's going to have an influence on your plant root stress. And so one of those things that we really want to think about is that plants are just allowing that temperature moderation. And then the questions become, okay, well, does that temperature moderation actually change yield? There are so many different components that go into yield that our research was not able to control that well enough. But our logic model kind of moves like this, where we have, all right, the cover crops lowered that soil temperature. That was measured, it was our observation that we measured. Does that lower soil temperature, we know that lower soil temperature decreases evaporation. That decrease in evaporation then increases late season water. That late season water we do know has an influence on yield. So our hypothesis could be that cover crops may increase yield. That is going to take a new set of research and some very elegant work to make sure that we are accounting for not only what's going on with evaporation, but also crop water use and the cover crop water use and transpiration and water infiltration in the first place. There's a lot of components of how we get from temperature to increased yield. But that's something that I think a lot of producers are looking for because they have heard over and over that cover crops are going to reduce my water. And then they see people who are having some really nice success with cover crops in non-irrigated systems, and they're wondering how that system becomes that water resilient system. And so we'd like to be able to move through that data. Okay, we're going to go to this next question from the audience, and that is, what is the optimal soil microbiology for a healthy orchard? And this answer isn't meant to be tongue-in-cheek, but I would say that optimal soil microbiology is one that is highly active, redundant, and well-fed. That is to say that there's no one specific optimal microbiology. And we'll talk about that in some of the data I'm going to share with you here. I have said high alpha and beta diversity. What that means is that there's a lot of different organisms and that there's a lot of different organisms in each group. That's what those different kinds of diversity measurements mean. Because one of the things we want is we want redundancy in these biological systems, because if one system, can't handle a certain stress, we want another organism that has that same set of enzymes or acids to be able to do the same job. We want different organisms filling those niches based on the conditions that are available at that time. Maybe one does it under high moisture conditions and another does it under low moisture conditions. And so when it comes to an optimal microbiome. We want them to be active, doing lots of stuff. The way that they do that is by being well fed. And part of the goal of having that highly active, well fed community is to have that functional redundancy. One thing that I'm just going to mention, it wasn't specific in this question, but I often hear people asking about an ideal fungal to bacterial ratio. And again, here I would say there isn't an ideal fungal to bacterial ratio. There are a lot of practices in ag that reduce the fungal communities. And a lot of different fungal species have a huge influence on nutrient cycling, water infiltration, flow, filtration, even water acquisition, and they work to help balance those diseases. So we want more robust fungal communities than what we get from many of the older agricultural practices. So oftentimes increasing that fungal to bacterial ratio is a good thing, but there's not an optimal fungal to bacterial ratio. There's not a certain number that I think is really a target when it comes to optimization. There's a lot of really good things that the bacteria and archaea and algae are doing as well. And so because we have had a history of not harboring a habitat for fungal communities, there's a lot of situations where increasing the amount of fungus can really have a benefit. All right, here's a fun data slide for you. This is something that I did from when I worked at the Soil Health Institute on the North American project to evaluate soil health measurements. So what you see on this map is a map of North America. Each of these black dots is where we had one of our long-term agricultural research experiments that we were able to sample. The color on the map is what we call the Hargreaves climate moisture deficit. So blue is there's lots of water, red is there's not nearly enough water. The small panel with all the little dots on the other side, each one of those dots represents the entire bacterial community composition from one sample. What you need to know in order to understand that graph is that the closer two dots are to each other, the more similar their overall microbial community is. So that means that if there's two dots that are bright red and right next to each other, they're probably from a similar type environment and they have a similar community. And if you see one that is orange and one that is blue and they're right next to each other, they have a similar community, but they're from very different environments. Here we see that there is a really strong pattern. And the title here is that inherent features influence soil's capacity. Because what I'm saying is that the climate has a huge influence on what that microbial community's capacity is. Okay. And so especially when we get extremes, when we get extremely wet or we get extremely dry, there are limits on what that microbial community looks like. So that climate has an influence on the community that might be there. Again, there's no ideal community. There's no ideal ratio. It has to do with where you are in the landscape. This is that exact same set of data, except this time we put different colors on the dots. And instead of having this Hargreaves climate moisture deficit, we have our pH scale. So we can see a really strong gradient that pH, the amount of hydrogen ions that are available in that soil, has a huge influence on what that overall microbial community looks like. In the study where we were looking at different orchard floor practices and hazelnuts, we had them kind of separated into our hill soils and our valley soils. And there were certain soils that were represented in our hill soils, Nakia and Jory soils series, and the valley was a Woodburn and Willamette. They have very different histories. Those soils developed under different conditions, different parent materials, and they had very different total soil organic carbon. And so when we were looking at the fungal community of these soils, what we found was that there was a huge separation just based on this landscape position. All of the soil, the key on this is that the triangles pointing down, that's the valley, and the triangles pointing up, those are the hills. So our hill soils are kind of all on this side, and our valley soils are on this side. There's a statistically significant difference between these 12 soils based on your landscape position community. And the cover crop didn't matter on this one. The landscape position was so much greater that this trial we couldn't see differences in the fungal community based on just the cover crop. One thing to think about is that landscape position, those hill soils had a lot more carbon. Part of the reason they had a lot more carbon is because they had a lot more clay. And those clay particles are going to hold on to that soil organic carbon. It's going to protect that clay in a different kind of way. And that has an influence on those microbes that are there. So this is the exact same picture as the one before. And you can interpret this similarly to what I was talking about earlier, where the closer two triangles are to each other, the more similar that fungal community is, right? So we have this one is colored by that soil organic carbon. And so these two soils both had very high soil organic carbon and are relatively close to each other. These two had similar soil organic carbon and are relatively similar to each other. So the reason that I bring this up is because it's really important for how we're going to track soil health improvements. These are some general recommendations. Number one, we want to be consistent. And we want to compare within your soil health sampling group. You want to be comparing within similar enough soils because otherwise your differences are going to be less meaningful. It's just going to be an impact of the soil.
Shannon, we have like
Yeah.
less than 5 minutes.
Yep. So we have our soil health sampling groups here. This is a map. I've got resources available for you, but this is based on ecoregions. So these black lines are ecoregions, texture. We have 6 different texture groups on here and 4 different drainage classes. And this is how we determine what your soil health sampling group is in order to do a fair comparison for soil health comparisons. And this is how we think about our interpretation. And that is that within the same soil, same ecoregion, we can look at something that has had no soil health management practices and something that has had all the soil health management practices. And we want to compare your sample within those bookends. So those are our bookends. This is a, saturated hydraulic conductivity, that outcome that we care about, the rate that the water is moving through the soil. It is really low where there's no soil health management practices. It's really high where we have all the soil health management practices, and where we've taken on some improved practices, it's in the middle. Similarly, we can see that same thing for what we call this minimum suite of soil health indicators. So if you're going to be tracking your soil health indicators, I would say this is our minimum suite. We use total organic carbon, CO2 respiration, and aggregate stability. And that carbon's going to tell us that total amount of food that's available. Respiration is the amount of work or the activity, because remember, they're exhaling this carbon dioxide. Every time they do metabolic work, they're exhaling CO2, they're respiring. And then aggregate stability, are they taking that carbon and are they building useful structures that are going to help us in our water and nutrient dynamics? I'm gonna skip over these lovely, lovely figures that I have, and end up here on how does the use of fungicides in my fruit orchard impact soil health? And I want to kind of bring this back to it. Number one, it's that use of fungicides. Yes, we're going to be killing some of the fungus, and that's going to have an influence on the other organisms in the soil. It does shift that fungal community. When we shift that community, it's going to change the soil's ability to perform these nutrient cycling, water holding, disease suppressions, and economic functions that we need for that soil health. I promised I would do one thing on diversity here. Just to keep in mind, most of the microbes in the soil are not described yet. We know about maybe 1% of them. The reason we need this diversity is because they need each other. They can't do this by themselves. That's why we can't culture them by themselves. So as we think about this diversity, we think about these systems, we can kind of have a little bit of humility that there's so much that we don't know. We're just getting into so much of this, and we can think about all of these organisms working together to create a whole society, like we have whole cells or we have a whole simulated environment. And so I am going to leave that one there, and then I know that Penny and Sue have a couple of closing thoughts, and I'm going to just put up the oh, that was my note on livestock integration. I know I said it earlier. We have an upcoming publication on considerations for contract grazing because it is something you need to be intentional about. So really think about that. And we have contact information and we have resources, and the whole slide deck will be sent to you for all of those who wish I could have had more time. Someday I will learn how to put the right amount of information in.
But Shannon, whenever you do a presentation, I feel like an hour feels like 15 minutes. I don't know about all the other attendees because I'm listening to everything and I'm like looking at the time, go, how is time happening so fast? So we wish you had more time as well. But maybe
Yeah.
just tell us just a couple minutes because it's so important about she'sorg.com because this is
Oh,
where you guys, the attendees, can go and be part of the programs and projects that you're working on and more questions.
yes, she's or is the soil health extension specialist for Oregon. So this is my new website. It is, it's just a couple weeks old, but there are lots of many resources. A lot of the information that I shared is on there. Things like our cover crop variety trial data reports are going to be there. There's a lot of different videos, different presentations that I've given that are up there, and ways to interact with us. So I want collaboration. I am an extension specialist. I want to talk with people. I want to hear your observations and hear your challenges and your problems that you want us to work on. And so, really importantly, I would say the best thing is if there's a question that you are unclear about, maybe it's something I can do a literature review on and provide that to you. Or maybe it's one of those things where, hey, we don't have a great answer for that yet. We would love to do some research. Maybe that's something we can do with you.
Awesome. Thank you so much, Shannon. That was so informative and really exciting, but also just how much that we still don't know. But some of that information I just found incredibly startling, especially the average temperature with cover versus non-cover, especially as we get hotter up here in Oregon. All right.
Yeah.
Well, Penny, can you share with us some places where people can get support? And I know that's a big part of what you guys do.
Absolutely. So I will go
Oh, yeah.
ahead and share my screen with you all. Alrighty. So whether you're already implementing soil health practices in your orchard, you're interested in trying something new, or you would like to connect with someone like Shannon to contribute to some research, we want to make sure that you know where to go to connect with that. And so if you're located in Oregon, the Oregon Climate and Agriculture Network manages a statewide soil health network which aims to connect farmers, ranchers, and farm service providers across the state, offering region-specific resources and connection to local support. Our goal is to ensure that all of Oregon's producers have the support that they need to improve soil health and build their climate resilience. Currently, we have 11 regional hubs located all across the state of Oregon. These are your local contacts where you can go to get connected with what support is available in your area, what local opportunities exist. Um, and they sort of serve as a one-stop shop because resources can be scattered, because they're different for everyone. Uh, these are your point people to connect with what financial assistance is available, what technical assistance you may need. It's a way to get connected with peer community, statewide resource people like researchers, and other extension professionals. And the goal is to make the experience of seeking that support a lot simpler for producers. On OrCAN's website, we have a whole page dedicated to our soil health network, as well as resources for soil health science and practical application. Encourage you to check it out. We will, of course, be linking Shannon's website under one of our many resources. Um, and you can join the network. Sign up at OregonClimateAg.org, take our network engagement survey, and you can receive updates about educational opportunities such as this events and get connected to your local soil health hub. And before we close out for today, I have some quick questions for you. So if you would take this quick poll. We'll give folks a few minutes to complete these simple questions. and embrace the awkward silence in the meantime. Okay. Thank you for coming out to today's webinar. Thank you to Oregon Organic Hazelnut Collaborative for supporting and helping to host this webinar. This webinar was also made possible through a grant from Western SARE and the USDA National Institute for Food and Agriculture.
All right, Shannon, Penny, thank you both so much for making this happen and providing such valuable information. And I know that for the hazelnut farmers out there, that the 2025 harvest was, as far as price per pound, the highest ever in our history. And with those additional financial resources, which doesn't come around very often, I think soil health is a great place to invest some additional resources. So this is such timely, valuable information. And so we'll be following up with everyone who registered with resources and the link to the recorded webinar and Shannon's information and Penny's information in case you didn't screenshot those. And then also, I just want to invite everybody to the Oregon Organic Hazelnut Summer Tour. It's our 9th annual summer tour. It's going to be on September the 1st, and we'll be following up with everyone on how to register. It's free, and we provide delicious food, and you're going to see so many cool things. We have a sheep grazing demonstration. We have a sucker slayer. We've got great presenters. We're going to learn about so many different things. So that's on September the 1st. All right. Well, we are on time, guys. It's 4:58, 2 minutes away. So I'll gift you guys a couple of minutes back and thank you guys again for being on the webinar today. And let's all come together and make our soil healthy.
Thank you, everyone. Thanks,
Thank
Sue.
you.